Thank you, Eleonora. Good morning, everyone. We are very pleased today to present you our work on generative AI. And today the title, as Eleonora mentioned, is Generative AI Under Pressure. The idea is to put in perspective how we use GenAI in real world use cases where you have many constraints in order to have a valuable output. So today the agenda is the following. We will talk about open problems. You know why GenAI is still not a solved problem. Then we will talk about new ways of developing GenAI technology. Then applying this GenAI technology in experiment to show and to compare versus existing one, and also the impact of those type of new GenAI technology on real use case. And finally, we will describe the new version of Micah, which is our de novo design technology, which allow to apply those GenAI in real life through a software. Through a software. I'm just trying to take the laser, the pointer, but it doesn't work. Okay. Anyway, I will use this. Okay. So first, problem number one. To be able to generate a nice molecules and to obtain a nice molecules, you need to fit a certain set of constraints. Because designing molecules is something which is very difficult. A good molecules, a nice molecules require a lot of parameters to be fixed to be able to be what we call a nice structures. And usually you start with very simple and classic physics descriptors. Then not only you want to have a good log-P, TPSA, you want to have good models in terms of solubility, stability, which can be global or local models. You can also want to have a good shape, a good docking score, so you can add 3D features. Then you have molecular features, precise features that the chemists want to fix, for instance, presence, absence of substructures, of particular motifs, spiroatom, etc., these type of things. Then all those constraints together, you are targeting molecules which are feasible, of course, but on top of being feasible, you want to be able to define a certain strategy in terms of chemistry, in terms of retrosynthesis, in terms of key intermediates. So as you can see, you have a lot of constraints to fulfill in order to have a nice structures. The problem is that the more constraints you add to the system, the more difficult it is to find molecules which satisfies all those constraints. And usually, one of the byproduct of addition of constraints is structural complexity. So this is something we need to live with. Then a second difficulty is balancing synthetic feasibility versus novelty. And that's very difficult because during many years, people prefer to work on virtual screening approaches because you control very well the synthetic feasibility. Those libraries are designed on purpose. But the counterpart of that is that usually you end up with low diversity because it's one reaction step usually with simple starting materials. So even if we are talking about billions of compounds, still you cannot control anything. The library is there and you have what you have. That's why in 2017, when GenAI first paper has been published, it was kind of a revolution because now you can control, you can generate whatever you want, 10 to the power of whatever. It's difficult to assess, but it's huge. So you can have high diversity, high control, but the drawback is that you have usually low feasibility. So can we find a middle ground where we can at the same time have a good synthetic feasibility and high diversity? And this is what we call at Iktos synthetically aware GenAI, new technology we are developing, which has the best of the both worlds, capacity of exploration, but at the same time, the capacity of generating feasible molecules. When you go through the literatures, Unfortunately, everything which has been published so far rely on smiles, graph, fingerprints, selfies, which are molecular representation, which does not take into account synthetic feasibility. They are not taking into account building blocks, synthetic transformation, or synthetic feasibility. So when you put those GenAI under pressure with many objectives, those GenAI start to hallucinate and create unrealistic and unfeasible molecules. That's a problem. So you're kind of trapped in a digital space where you can never go to the lab because you cannot make those molecules. So what's really set Iktos apart regarding the crowd here is that we are the only company which has been able to produce a chemistry-driven, synthetic-aware GenAI technology. The problem, as I said, is like large language model, GenAI based on word. They don't know nothing about chemistry. So they are just playing with permutation of characters, permutation of piece of smiles, and at the end of the day, generating a new smiles with permutation, of course, can generate new molecules, maybe novel parts, but the drawback is that this is not feasible part. Contrary to reactions-based GenAI, which combine building blocks, reactant, reagents, and reaction, name reaction, allow you to build by design a molecules, which is a consequence of steps which are synthetically realistic steps. So what chemists need, of course, chemists used to use during many years virtual screening, which was a good approach at that time. Right now, GenAI is totally surpassing virtual screening with the problematic of low synthetic feasibility. That's why what chemists want is, of course, GenAI, but in a space, what we call synthetically tractable space, where you can, at the end of the day, make the molecules. So you are looking for molecules which are synthesizable. You are looking for molecules which maximize the constraints of your project. And of course, you're looking for something novel. And we believe this is where our GenAI can really bridge the gap between virtual screening and black box generative AI. On top of that, of course, chemists want more that feasible molecules. They want to be in control. They want to select the exit vector. They want to be able to select the catalog of the providers, the chemistry involved in the process. That's why it's important to have this capacity of control. And I will let Stephanie present it how in Makia we can control that.
Yes, thank you, Quentin. So now that Quentin already presented the issues and the problems that we are facing in the industry for the early stages of drug discovery, now I would like to talk a bit more how we rethink GenAI for chemistry, and more specifically, how we enable chemistry-driven design strategies using our platform. So unlike any other typical GenAI models that you just generate smiles or optimize black box scores, our GenAI is basically built to use chemical logic. So it understands structures, reactivity, and constraints at the building block level. So this means that we can ask chemically meaningful questions, for example, in the presence of a reference molecule like the one that we have here, where we want to develop new ideas. The kind of questions that we can ask are like, what if we replace the R1 moiety with synthetically accessible alternatives? Or what if we substitute the head group R2 using specific name reaction? Or finally, what if we explore new heterocycles by hopping out the core and the substructure constraints? So to answer the question if GenAI can use chemical logic to generate new ideas, the answer is absolutely yes. So specifically, our GenAI enables two chemistry-driven design strategies, which are growing new molecules and linking fragments into novel compounds. So in growing mode, we start from a non-substructure and selectively expand it. And then on the other hand, in linking mode, we bridge fragments to generate synthetically viable compounds. So here's a closer look at how that works. On the left, you have our GenAI, where we are growing molecules step by step, using real reaction templates and respecting reactive constraints. On the right, links fragments by proposing realistic reaction-compatible connection, and it follows the same logic as the fragment growing on the left side. So the important message here is our GenAI is grounded in how chemists actually design molecules. This is where our technology really separates from the standard GenAI. Every transformation you see here is grounding in reaction reality. Our GenAI does not hallucinate. It works with building blocks, it applies validated reaction templates, and constructs synthetic pathways that are actually usable. So it's a generative system, I would say, that respects synthesis by design. So if you want the full details, we laid them out in our preprints. So here we break down how the growing and linking optimizers actually work, how the models are trained, and how the architecture mirrors chemical logic from reaction selection, building block selection to retrosynthesis tree. So in the previous section, we analyze how GenAI can effectively be used to allow in silico chemistry experimentation. However, what about synthesizability? For that, we need to review how our algorithm actually works. So as stated before, our GenAI doesn't just guess molecules. It builds them one step at a time with synthetic feasibility at every decision point. So our GenAI starts by searching 1 million of commercially available building blocks using AI-guided prioritization. So basically, it picks the best one to begin with the synthesis. In this specific case, it's a protected spiroamine. Then the next step is predicting the most suitable reaction type from over 2,000 organic transformation, predicting the optimal reaction. That first building block and reaction, they yield viable intermediates. So in this case, a BOC2 protection to unlock a key amine. Now the process repeats. Our technology is able to go through another round of selections of building blocks and reaction templates based on these created intermediates. So this builds the final virtual synthesis compound. And so here at this point, it's important to clarify that our AI can go up to 4 steps, meaning that these steps that we just discussed here can be repeated 3 more times. So the important message here is that this is an in silico synthesis planning, but with chemical intuition built in. So our GenAI doesn't just propose ideas, it proposes also how to make them. So after discussing in silico experimentation and how we tackle the synthesizability issue, now it's also important to discuss how we optimize the generated molecules towards the objectives of your projects. So at the heart of our generative AI is optimization using reinforcement learning in order to incrementally converge towards good molecules that are predicted to have strong properties. So why reward functions are actually important? Because they are essential to detect undesirable properties early, guide the AI in refining substructures, prioritize promising candidates, and also enable efficient multi-parametric optimization. So the possible reward functions that you can use with our GenAI are, for example, the Tanimoto similarity to a reference space. So basically here, if you have a chemical series, or maybe you have a compound of your design that you want to continue exploring, but not moving very far away from your scaffold, then you can use this constraint with our technology. Then let's say that you also have your 3D structure base. So you have the crystal structure on your protein and your reference ligand. Then you can use the 3D structure base optimization in our loop. So here what we optimize is docking score and interaction fingerprints. So the interaction fingerprints are actually very important. It's a very important reward because here it's a way that we have to tell our platform which interactions are actually essential for you and what interactions you want to absolutely have in all the generated molecules over any other interaction. So in this way, our platform will generate molecules featuring only these interactions that you are interested in. So then if you have in-house models that you also want to use to score molecules using our platform, it is also possible through an API. And so this way, our platform will use your information to score your molecules and optimize them. Then, if you have activity data or ARMI data, then we also can give you the possibility of building local models in our platform. So in this way, you can optimize molecules that are optimizing the targets that you're interested in. And finally, if you do not have your 3D structure base, but you do have the information of your ligand, then you can work with the 3D ligand base. And here, basically, what we do is we optimize the shape similarity and the pharmacophore. So here I want to mention an important point. All these reward functions for the multi-parametric optimization are fully based on user-defined rewards. It is actually you, the user, the one that is going to decide which rewards you want to use for your projects. So building on the previous concept, once we define the target product profile, the real work actually begins for our GenAI. So here we start with our GenAI, which designs novel chemical structure. But this is an unconstrained generation. It is grounded in real world chemistry. The AI pulls from a library of over 1 million commercially available building blocks, more than 2,000 well-established organic reactions, and multiple reaction steps. So in other words, every molecule proposed is by design synthetically accessible. Then we enter the optimization loop. So these generated molecules are then scored using reinforcement learning based on how well they meet the target product profile criteria. So these scoring systems act like a reward function, guiding the AI to prioritize more promising candidates in every iteration. So the result is a tightly integrated chemistry-aware design cycle that doesn't just generate ideas, it delivers optimized, synthesizable molecules, and that also align to your target product profile in your real-world project's goals. So I will give the word now to Quentin back.
Thank you, Stephanie. So yeah, so we decided to bench our GenAI versus reInvent, which is a very well-known GenAI, open source GenAI, and we believe that it's one of the best one. It's supported by the community, but also it allows to grow fragment, to link fragment, to perform some fine-tuning. So it's quite comparable to what we are doing. So we thought that reInvent for it, it's a good benchmark. And we decided in that paper to try real use case at the same time, but also different use case you may encounter during your project. The first one is trying to recover non-structures. So here we have 8 molecules which has been published in the literatures, some with some macrocycles, some with complex scaffold, or with stereocenters. So we say, okay, what happened if we ask the GenAI to reconstruct those molecules by only following and using Tanimoto similarity as a fitness function? So what we did is we performed 3 different types of experiments. The first one is we do not give any details of starting material to the GenAI. It has to start from scratch to reconstruct using Tanimoto similarity the targets. The second experiment was with a fragment. So you start with 1 fragment of the molecules, and the third experiment was to use 2 fragments and to see if it's able to rebuild the molecules by finding the good one in the middle. As you can see here, when you reach a Tanimoto of 1, similarity equal 1, it means that you have been able to find back the expected structures. And as you can see here in pink, Iktos is very often able to reconstruct actually the target molecules, where for reInvent, it's much more complex using this GenAI, and it's always, in most of the case, below Tanimoto similarity compared to Iktos. For instance, lorlatinib, which is a microcycle, reInvent is not able to generate any macrocycles, so you get stuck to a very low similarity. In contrary, our GenAI is able to reconstruct this macrocycle by a simple Tanimoto similarity. Another benchmark, which is also very interesting, is more a lead optimization benchmark. In that case, we use a data set of almost 500 molecules published by Boehringer Ingelheim with molecules tested on PI3K and mTOR. And in that case, we build QSAR models on those objectives, and we try to generate molecules, new molecules, maximizing those two endpoints simultaneously. So the design objective were QED, which is drug likeness. Then we wanted to have a Tanimoto similarity higher than 0.5. We wanted to have a good PI3K mTOR prediction using QSAR model again. We wanted to maintain this part here as a scaffold. And in post-processing, we compute retrosynthesis to assess the top 500 molecules generated by MACHA, generated by reInvent, and to compare the synthetic feasibility, synthetic quality. In case of MACHA, we wanted to start with the chloro, which is a logic disconnection in chemistry to react in this position. And in reInvent, you need to put a star because reInvent is not chemistry driven, and we compare the output. So the output is very interesting because if you take the top 500 molecules, Iktos produce 85% of feasible molecules with a score higher than 0.5, meaning that it found reasonable retrosynthesis tree compared to reInvent, where only 50% of the molecules looks feasible. If you have a look as well to the diversity, our GenAI produced much more diverse compounds, almost 3 times the number of new scaffold and different scaffold compared to reInvent. Also, if you check the scaffold similarity between each other, we have a lower similarity between each scaffold, meaning that we have a good diversity. And the same for molecular similarity, we have a lower molecular similarity. So it shows that even using QSAR model plus Tanimoto similarity, this type of GenAI, which play with building blocks and reaction, allow you to get very diverse structures compared to GenAI playing with characters permutation like reInvent. Also, we wanted to apply those GenAI in 3 different projects, following and maximizing docking scores, so PIM1, ORC2, and TMRD, which are 3 targets where we have the structures from the PDB. Then we use Autodoc VINA to maximize binding energy, and also Iktos contact score, which is 3D interaction fingerprints in order to be able to reward some amino acid you want to interact with. In those three examples, we start from fragments, and we decided to grow in certain direction of each of those fragments, maximizing 3D scores. Interestingly, in the PIM1 example, reInvent appeared to produce interesting results. The top 500 molecules were almost all feasible. But in case of ARC2 and TRMD, the results are much are less good because only 100 molecules appear to be feasible in that generation. And here, half of the molecules appear to be feasible, contrary to Iktos GenAI, where in all cases, we obtain almost only feasible molecules. If we focus on ARC2, it's interesting that we started obviously from this pyridine. So we have the pyridine everywhere, and we have different scaffold, sometimes also different branches. We have a very good score of 0.9, almost 1. So very good in silico scores, contrary to reInvent, where the system has started to hallucinate, generating this type of complex macrocycles, a lot of aliphatic ring, so producing maybe scores at 0.8, almost 0.9. So in silico, they look good, but when, as a chemist, you have a look to the structures, you don't want to synthesize anymore. Also, in terms of diversity, it's interesting to compare the pairwise Tanimoto similarity on those 3 examples, where we can see that we have 0.4 pairwise Tanimoto similarity, 0.22 and 0.39 with Iktos GenAI, which again produce very different molecules from each other. and high number of new scaffold. Also, if you perform a t-SNE on the generation, for instance, on ARC2, you can see that the reinvent molecules in orange are all spotted at the same place, contrary to Iktos one, where it produces a wide range of scaffold and structures. So again, we are totally convinced that navigating into the space of building blocks and reaction allow you to perform some big jump into the chemical space, contrary where when you permit characters, you perform very small and tiny modification in that space. So not only you produce less diversity, but also you may produce some totally unfeasible molecules, because just by changing one character in your smiles, you can go to a totally feasible molecules to one which is completely unfeasible.
So I'm taking back now. Thank you, Quentin, for presenting this last part. So now that it's time to put everything together, because we talk about the problems, we talk about how we approach also the issues of synthesizability, and how we also allowed chemists to work with all the chemistry constraints that they want to in our platform. So now let's talk about the chemistry-guided de novo design of molecules and how you can use the optimization of these reward functions as well. So our GenAI isn't limited to a single task. It is built to handle a wide range of drug design challenges. So from scaffold hopping and heat expansion to lead optimization with multiple objectives, we cover the full spectrum, all while ensuring synthetic accessibility, 3D constraints, and multi-parametric optimization align with your target product profile. So this is a modular, flexible platform designed to tackle real-world constraints and not just generate molecules, but also deliver viable candidates. So using our technology, you can have access to multiple paths to generate molecules. So if we start from this reference molecule that we have here, then you can generate novel ideas from different starting points of the molecules. So the first one is that you can grow new ideas starting from an initial fragment in one direction. So for example, here we are using the bisfluorophenyl here and the boronic acid as the reactive center. And in this way, our GenAI will keep this scaffold and then will introduce diversity in the rest of the molecules, as you can see in these example molecules. So also these molecules are generated guided with our 3D structure-based constraint, which are docking and contact score as previously mentioned. Then the second thing that you can achieve using our algorithm is to also grow new molecules, but in this case into different directions. So the logic here is the same as the previous one. But now instead of growing the molecule in one direction, we are doing so in two different ones. As you can see here, the final molecules, for the final molecules, our GenAI will keep the core of the initial fragment and then add diversity into different directions. And once again, these molecules in this generation were also optimized against 3D structure-based rewards. So then the third use case that you can achieve with our technology is that you can link two fragments. So in this case, instead of starting from one initial fragment, you're going to start from two of them, and then you will generate diversity in between. So in this case, as you can also see here, we have the two initial fragments, which is the boronic acid again and the cyclic amine, and then our platform will introduce diversity in between them. Then let's say that you're interested about only generated microcycles, then it's also possible to achieve it with our technology. So here we are starting with two reactive centers, which are the bromine here and the chlorine here. So what we are going to ask our technology is to generate only microcycles starting with these reactive centers. And in this case, we use the 3D ligand-based reward, which are 3D shape similarity and pharmacophore to guide the optimization of molecules. And so finally, we can also use our technology to fine-tune. So this is more used in the lead optimization stages, where you have already a chemical series which is defined, and you already know which is the chemical scaffold that you want to explore. But you want to maybe increase activity or maybe certain ADME properties, staying close to your chemical series. So this is also possible with our platform. And as you can see here with our technology, we can start from the common scaffold of your chemical series and then ask our software to give small modifications in the rest of the molecule in order to increase certain reward properties. So in this specific case, we use QSAR and similarity as reward functions to make sure we optimize molecules, maximizing activity and ADME models, and also staying close to your initial chemical space. So as I mentioned earlier, our platform is unique in the way it leverages AI driven by chemistry and guided by chemists to make synthesizable compounds. To wrap it up, on one side, we have our GenAI, which performs the de novo design of synthesizable molecules. On the other hand, the AI-driven photosynthesis technology finds the best synthetic route. As we discussed before, our approach to generative AI goes beyond simply generated molecules. Our target is to generate synthesizable molecules to accelerate the DMTA cycle. So to do so, we work with a data set of 1 billion building block and 2,000 named reactions. And in this way, we are incorporating chemistry in our GenAI technology. So we leverage the power of chemistry information to synthesize molecules molecules which are synthesizable by design. And then this molecule can be optimized against the multi-parametric optimizations that we discussed before. So I want to also insist once again that this is a user-defined part, the multi-parametric optimization of the reward function. Because at Iktos, we strongly believe that AI will not replace chemists. Human expertise is essential to define your project's requirement, to choose a strong setup, and eventually to select interesting results for further analysis. So finally, our AI-driven photosynthesis software helps chemists to prioritize those molecules with feasible routes that fit the requirements set by the med chemist or synthetic chemist and optimizing cost, time, and complexity. So I will give back the word to Quentin because now we are gonna talk about 2 use cases to illustrate how we use our platform.
Thank you, Stephanie. So on the following slide, we decided to test our GenAI on a target called PKMYT1, which is a kinase from the WEE family, which regulates CDK1 and which has a very important role in DNA damage response. And it's a field which is quite crowded. There are a lot of people working on this target. One of the main actors is Repair Therapeutics. And they have the non-receptive in phase 1 right now. As you can see, it's interesting. We can see the structure here in 3D into Machia. And we can also define some score we want to maximize. So here we want to make interaction with the hinge binding motif. We also want to have high stacking on the central scaffold, etc. So you can define that. And what's interesting when you see non-receptive is that you have this nitrogen here in the central scaffold. And also they produce several patents with always the same type of motif here. When you see the competition is the same, then Atlas, Simres, Insidico, Chugai, Excelixis, etc., all those companies have the same pattern. So you have on one part a phenol, on the other part some sort of an amide bond to interact with the hinge binding motif. And in the middle, you have NH2 everywhere. Maybe one exception is these molecules from Insidico. So we ask ourselves, can we use our GenAI to produce something totally different than all those scaffold and see how it goes and see the activity. So this was the goal of this generation, being able to produce something which does not contain this NH2 moiety. So for that, it is very simple. You just put your PDB file into Machia, and then we wanted to start from this phenol, which is our starting point, which set in the back pocket of the target. And we asked Machia to generate something different in this part of the space. So the starting point were the following: either a bis methyl or a monomethyl with a bromide to perform some sort of coupling, or with boronic acid. We wanted at the same time to interact pi stacking with some amino acid, pi hydrophobic, H bond, etc., in order to maximize key interaction, keeping similar shape, but obviously having a totally different scaffold. So in several hours, you can do that into Makia. Then you can make your selection and you can synthesize the compounds. Makia produce 30 totally new scaffold, and we decided to select 16 of it and to synthesize those scaffold, those molecules. Out of those 16 synthesized molecules, we get 4 active molecules from 600 nanomolar to 13 nanomolar activity. So very high active compounds in only 16 synthesized molecules, and 4 with totally different scaffold. And this one is quite unique, never described in the literatures, and no NH2. So it allows really to obtain something totally different and to have different type of physical properties. It's interesting as well to mention that those molecules has been produced on our robotic platform thanks to our orchestration technology. So, and in 2 reaction steps in average. So you don't need to do a lot of chemistry to find something very active and totally different and novel, because here it's clearly what we have been able to obtain. Also, another approach, we decided to use the fragment linking. So we keep the phenol on the left, we keep the amide bond on the right, and we wanted to find something new. In that case, the same left-hand side, but for the right-hand side, we use these derivatives. We keep the same interaction in place we wanted to keep. And in that case, we synthesize 24 molecules, and we identify 3 active scaffold. Again, new scaffold, no NH2. In that case, we decided to keep the amide bond, which was not the case in the first experiment, and we obtained 3 compounds, less potent than the previous generation, but again, in only 24 molecules, we obtained 3 active compounds with totally different scaffold, totally different physical properties. So in total, in less than 40 molecules, we have been able to identify 7 active scaffold with 7 different types of properties, which allow the team and the chemist to follow different strategy for that project. And since we are not able to show you the structures on those use cases because it's our in-house pipeline, Stephanie will show you a use case with GLP-1R where we can show you the structures.
Yeah, absolutely. So, so this use case highlights, I would say, the kind of problem solvings that you can achieve in Makia when working with challenging target like the GLP-1R transmembrane protein. So, just for introduction, GLP-1R, it's a particularly important target because when activated by its natural agonist, GLP-1, it triggers a G protein signaling cascade that leads to insulin release and glucagon inhibition. So this makes it very important targets in treatment of diseases like, for example, type 2 diabetes. So there are several peptides-based GLP-1R agonists already on the market. However, there's a growing need for new small molecules agonists. So in this use case, we decided to work with the Pfizer compound Perron, which was recently discontinued. However, it's shown promising results, and we can use it as a starting point to design new chemical scaffold. So the initial success of dinolipron can be largely attributed to its ability to engage deep within the receptor binding pockets, specifically through water-reaching interactions with residues in the transmembrane using the carboxylic acid. Also, the oxetane moiety forms important hydrogen bonds that contribute significantly to the compound's potency. So with this in mind, if we want to design new compounds using dinolipron as the reference ligand, it is crucial to retain both the carboxylic acid and the oxetane as starting point. So we applied here a fragment-based approach to generate novel molecules using these key functional groups as the co-element. So through a fragment growing algorithm, we will introduce chemical diversity while preserving critical interactions. So starting from the previous initial fragment, fragments, we target the generation of novel chemistry in the rest of the molecule. And so this strategy allowed us to explore interactions that are already present between the reference ligand and the protein, that like the one that I'm showing you on the left, but also explore new interactions with residues like, for example, the lysine 217, allowing us to generate a more diverse set of molecules. So now that we have identified our project's goals and we have identified the initial fragments, it is possible to start with the generation. So from our software perspective, we have our initial fragment, which contains the carboxylic acid and the oxetane, and then we're gonna ask our software to grow starting from here and adding diversity. From the point of view of the reward functions, we use two of them, which are local models, activity models, and then we have structure base. And also it's very important for you to understand at this point that you don't need to use them one reward function and then the other. You can couple them both at the same time in the same generator. And that's what we did in this use case. So just keeping the previous initial fragment, here I want to illustrate the different strategies that you can work with when thinking of different kind of chemical structures that you want to observe. Like, for example, if you're interested in just expanding the molecule and exploring the hydrophobic pocket, like for example here, where we also have the new interaction of lysine 217, then you can start, for example, from an apipyracine and ask our software to come up with reactions and building blocks towards starting from this amine. Then, if we want to explore more in the middle of the pockets, where we have, like, for example, a lot of pi interactions, we can start from another reactive center, like in the second example. So here I will show you some of the results that we got home, from Makia, and so you can get with our platform. So depending on the starting point, the reward function, in this case we use different interaction fingerprints, and the reactivity that you get in your initial fragment, because the reactivity here is not the same as here, then you'll get different interesting ideas. So it's interesting to observe that here, for example, in the ring closure, you can go through probably a benzene midazole synthesis using an aldehyde as building block, and also these aryl amines here to close the ring, and then probably some aldehyde reductive amination. So here the strategy to demonstrate actually that Makia doesn't just generate trivial reactions. It is also able to perform more complex reactions, such as decyclization in the example of the ring closure. So then I just wanted to show you that our platform is also able just to dock your molecule within the protein pockets and to give you the interactions. This is an example of a series that we've continued working with these generations in mind. So as you can see here, I'm comparing the reference ligand with the molecules that we generated. Of course, these molecules, they are not decorated at all. It's just a scaffold because we wanted to explore this at the beginning and to see what kind of interactions we had. Okay, so I know that this is a topic to really discuss in more into detail. That's why I invite you to go into this demo page. You can scan the QR code, or you can redirect into the link that Eleonora will send you. And so here you will be able to see the full GLP-1R use case with the settings in our softwares and with a more proper results of all the structures that we've generated. But also you will have publications and other demos where you can get a better idea of our software. So I invite you to visit this page. And just to close and to finish the webinar, the last topic that I want to discuss is that, of course, after you generate molecules, you need a tool that helps you to prioritize only those synthesizable low-risk molecules. So now we are putting a lot of effort developing a new software, or a new version of our software, which is Makia 2.0. And here what we are hoping is, apart from being able to generate molecules, it is also to give you the tools in order to analyze your results. So as you see here in the screenshot, you will have different kind of ways to explore your results and to prioritize the molecules that you want to keep. So once again, I invite you to our demo page, because here you will find a small video in where you will have a preview of what's about to come with respect to our software. So before we jump into the Q&A, I just want to thank you all for being here and for your time listening to us. And so I will give back the word to Eleonora so we can answer all your questions.
Thank you very much, Stephanie and Quentin, for the really interesting and compelling presentation. We will start now the Q&A. Please remember that you can type questions in Japanese in the chat, and we will invite our colleague Misuki for translation. So we will take the questions as we receive them. So the first question is for you, Stephanie. It says, Stephanie said about 1 million in stock building blocks. How many suppliers did you gather those building blocks from, and are they all affordable?
Okay. Yes. Okay. As of today, we have around 40 different suppliers. I cannot tell you that all of them are going to be affordable, but what I can tell you is that we have filters in our platforms in order for you to indicate the price of the building blocks that you want to work with. Let's say that you don't want to work with building blocks of more than 100 US dollars, then that's a constraint that you can introduce in our platform, and then we are gonna give you molecules, the post-processing, where you can prioritize all those building blocks that are not expensive. Yeah.
And I think so far we reached 40 different type of providers.
So let's continue with our second question. What is the optimal way to utilize Iktos tools when X-ray structural data is not available and limited information on activity is present?
So
if you want, okay.
Yeah,
okay.
You can take it, Stefan.
Okay. So when you don't have the X-ray structural data, you can decide if you want to design your molecules in 2D or 3D. You can do it both in our platform, but we have also the option of 3D ligand base. So here, it's either you have the conformation of your reference ligand, or you don't have it. It doesn't matter, because if you don't have the conformation, then we can generate it for you. And in this way, you can guide the generation with, for example, the 3D shape similarity and the pharmacophore. And also, if you have a limited activity data, then that's not a problem. If you have, if you want to design at least diversity at the very beginning, and then since this is an iterative cycle in which you generate molecules and then you synthesize and you get information, then you will have this information with time, and then you can build your system. That's actually the targets that we want to achieve with our software.
And in case you have absolutely nothing, so you don't know the ligand, you don't know the targets, as you may know now, there is a BOLS2, which is an open source package published by MIT and Recursion, which allow you to start from a sequence of amino acids. And this is something you can also plug through API to Micah to generate from scratch using only a sequence of amino acid, a new generation.
Thanks a lot, Quentin, for the clarification, because that connects to a question we also receive on how to address cases where you're working just on a very novel target. So we will continue answering the questions in the order we received. So I think we're changing a little bit the tone, but it's good to mention how our tools are available. So the question is, are Iktos GenAI tools downloadable software, or is there a GitHub link to follow in the research? So I think maybe it's good to mention how Spire is available.
Yeah, so our software are not downloadable because they are computationally intensive technologies. It has to be connected to docking. It has to be connected to GPU to use GenAI technologies. So right now it's available through a SaaS mod, software as a service, which are usually deployed on AWS, either Iktos AWS or our client AWS. It's the same for Micah and Spire, it's also publicly available. So if you want to have a look to our retrosynthesis technology, you just need to go to our website and to go to Spire and to create an account.
So we move to our next question. To do scaffold hopping on a particular part of a molecule and to find a similar bioisoster, is that possible? And is it possible to design dual inhibitors using PDB codes at the same time?
Yeah, so the notion of bioisoster is difficult. So it depends. What I can say that by generating something with a similar shape and making similar interaction in 3D in the pocket can be considered as a bioisoster. So yes, it's possible actually to pattern bust, to find something different. And also right now in Micah, you can upload only one PDB at a time. So you can apply the generation using only one PDB at a time. If you want to build a dual inhibitor, you need to set up an API. So in Micah, you will generate maximizing one PDB file. Through the API, so Micah generating molecules, and you can use external API to score those molecules. You can also put in place another docking score for the other PDB file. So it will require a bit of setup, but it's doable theoretically.
Thanks, Quentin. So now we have two questions that have been asked in Japanese, so we will invite Misuki to the stage. If you can join us, thank you, and translate.
So the question is, can we specify the size of the core structure as well as the total number of rings within the generated structures?
Stephanie?
Yeah, you can. Absolutely. We have control at the building block level and at the product level. So you can specify the number of rings that you want to have and the core structure. Like, for example, you can specify the molecular weight, or you can specify the number of rings, aromatic rings, aliphatic rings that you want to work with. But that's not the only thing that you can do. You can specify the number of hydrogen bond donors, acceptor, TPCA, QET, and all kind of descriptors that you can imagine from the physical chemical point of view. And also you can specify constraints from the structure point of view. If you have smarts or if you have substructures that you want to keep during the generation, you can specify it either in the building block or in the product level.
Thank you, Stephanie. 母核のサイズや間の数を指定することはできるのでしょうかという質問についてですが、可能です。ビルディングブロック、追加するビルディングブロック、もしくは最終的に生成される化合物に含める、間の数分子量の、上限下限を設定していただくことも可能ですし、その他にも、アロマティックリングをいくつにするのか、アリファティックリングはいくつにするのか、ハイドロジャンドナーボンド、ドナーとアクセプターの数はどうするのかなど、さまざまなデスクリプターに、の上限下限を設定することができますので、ご質問への回答としては指定可能でございます。I'm done.
Thank you, Misuki. Maybe we can go to the next question that I was asked in Japanese.
Yes. The second question is around SP3 character within the building block to be added. So the question is, my understanding is the SP3 character will be poor when building blocks with high synthetic feasibility are added. While a fragment with high SP3 character is thought to be difficult to synthesize, it will indeed increase the diversity in the 3D structure as well as the binding affinity for the target. Additionally, fragments with high 3D character are believed to improve the physiochemical properties like solubility. How does GenAI from Iktos deal with this point? Is it less likely to generate structures with high SP3 character?
Yeah, that's a very good question. In our library, we have a lot of SP3 rich building blocks. We are working with an amine. We also have access to Spirochem library. So which is very cool is that at the same time you can have SP3 rich generated molecules, but easy to make because we buy directly from providers the building block. So we have the best of both worlds, SP3, but still feasible thanks to our providers.
Thank you. こちらのご質問への回答としましては、特にビルディングブロックの SP3 性 によって何をマキアに入れて入れないのかっていう選別はしておりませんので、基本的にプロバイダーさんからいただいたカタログのビルディングブロックはすべて入っている形になります。そのため特にSP3 線に乏しい構造が出てきやすいなどということはなく、ユーザーが設定したコンストレインに沿った分子が出てくるように作っております。Okay.
So we will continue now. The next questions are asked in English. So I will go ahead and read them. Can your generative tools be used to identify improved ways to synthesize a given compound? I'm thinking about potential applications in process chemistry.
So yeah, the GenAI can generate molecules. So it's not really related to retrosynthesis. I think in this case, it's better to switch to SPIA. And in that case, yes, in SPIA, this is really the goal of SPIA, is to find all the possible routes to make a molecules. So you could find, using SPIA, a totally novel route that has never been published before. And in SPIA, you can also put constraints in terms of price of the building blocks, chemistry you want to use, chemistry you want to avoid. So somehow you can customize the retrosynthesis search to focus more on process chemistry.
Thanks, Quentin. Then we have a very short one asking whether the platform depends on published Python libraries.
Yes, yeah.
I guess that you're talking about like more open source Python libraries. So we have our own code, of course, but we also we can use open source Python libraries, either that they're already within our software, or if you want to apply your own, you can connect them through APIs into our software as well.
So Iktos GenAI is proprietary GenAI, and then of course we use Python library like RDKit or
RDKit,
but all the generative aspect and retrosynthesis aspect is proprietary.
Thank you. So now we're going back to the point of the building blocks. Are 1 million building blocks are all commercially available?
Yes, they are all commercially available, and we curated them because actually we have 60 million building blocks that we used in our SPIA AI-driven retrosynthesis. By in Makia, we curated them with respect to different kind of physical chemical constraints. So they are really proper building blocks for drug discovery and chemistry in general.
Thanks, Stephanie. So now I think this can also be for you. We have a question concerning the GLP-1 case that you presented. They say that they might have missed this, but in your GLP-1 case study, what was the potency compared to the starting point? So maybe to clarify.
I wish I could give you a number, but we did not synthesize these compounds just yet. Because yes, as I said, here we are exploring more scaffolds and not the fully decorated molecule. But maybe at some point we will be able to do it, and we check. I'm sorry that I cannot give you a number.
Yeah, but maybe good to mention that the recordings of that use case, if you want to go through it, are in the link to the demo page that we just provided on the chat. So you can find more details. The next question is in Japanese. So Mitsuki, if you can translate, please.
Yes, the question is, please tell us more about Iktos robotics platform.
That's a good question. It would take 1 hour, but it's good because for the next webinar, we will focus more on the robotic aspect. But in few words, basically, Makia generate molecules, SPIA find routes, and then behind we have ChemSpeed, which is piloted by our AI technology called ELACA, in order to transform a list of molecules to instruction for the robots and to execute automatically the chemistry. So just register to the next webinar and you will be able to see more about our robotic platform.
Thank you, Quentin. 次回のウェビナーのトピックがロボティクスの予定ですので、ぜひご参加いただければと思いますというのが回答になります。で、簡単にご説明させていただきますと、マキアで分子を生成した後に、その生成した分子のリストがイラカという、 プラットフォームを伝ってロボティクスに行くんですけれども、そのイラカが AI テクノロジーで、そのリストに含まれている分子をどういうふうに、ロボって生成するかっていう、このトランジションのところにIktosの AI 技術が使われています。詳しくはぜひ次回のウェビナーにご参加いただければと思いますので、お願いいたします。Okay, let's move on.
Thank you very much, Mitsuki. I need to point out that we are on the time, but we have many more questions to answer, so we will continue a few more minutes to keep the discussion. So the next question is about prediction models. Do you typically use AI prediction models when generating molecules with Maca 3D, as in the case of the GLP-1 use case?
So that's a tricky question, because predictive models and 3D structure-based, you need to know how to balance them, because of course this is still an algorithm, and so if you're feeding a lot of information that they are contrary, like for example to optimize something in one direction and another thing in the other one, then the algorithm won't know which is the proper molecule. But just as a good way to go, I would say I would use the 3D structure-based and activity as the first starting point, just to optimize things. But I wouldn't go beyond that. I would not use a lot of predictive models on the top of the 3D, because also you are kind of like, it depends on where you are in your project, but if you are using a lot of predictive models with the 3D, maybe you're missing or you're losing diversity. So it's something also that just to have in mind how to properly use them. So my recommendation, or the way that we'd like to work, it's like 3D from one side and then activity and see what happens, but not more than that. That's what I would say. Yeah. Then for the multi-parametric optimization, that's the case that Quentin presented in our publications. There, for example, we optimize like 6-7 targets without the structure, because of course we are in another stage of the project.
Thank you, Stephanie, for the answer. Now we're changing a little bit the topic to deployment. Can the software be also deployed on other cloud platforms apart from AWS? And what if a company uses other platforms?
So right now we stick to AWS.
Thank you. Mitsuki, if you can do the translation.
Yes, the next question we got is, can we register our in-house building blocks, and also can we specify which types of building blocks we want Makia to consider?
Yes, everything is customizable. This is usually the first thing that the customer asks, to add his own inventory. And in Makia, you have the option to play with that and to select the one you want to use.
ご質問ありがとうございます。自社のビルディングブロックの追加は可能かというところと、登録されているビルディングブロックを選択して、文章を生成させることは可能かというところなんですけれども、どちらも可能です。自社の、ビルディングブロックリストをこちらにご共有いただいて、御社のカスタムマキアをご提供する形になります。あとはすでに登録済みの、約40社からのカタログ情報の中からビルディングブロックどういったものを優先して使っていくかっていうのもジェネレーターを設定いただく際に選択いただくことができます。Okay, next question, please. It's me.
Yes, we have 3 more questions, Mitsuki.
Okay. The next question is, does Makia give us synthetic roots? Maybe it gives us only structures whose synthetic routes can be easily guessed.
I can take that one if you want, Quentin. So Makia does not give you synthetic routes, but with this being said, we give you like the way that the algorithm think to give you the final molecule, which is not the synthetic routes, but at least we give you the building blocks that the algorithm used for the final molecule. But also, when you generate molecules using Makia, then you will have the opportunity to click on the SPIA button, and if you have both, you will be redirected to this page where you will have displayed all the synthetic routes. And even if we don't give you synthetic routes in Makia, we give you our score, which is synthesizable score. which are customizable from your side. And so maybe you can include intermediates or building blocks that you want to work, and it will be easier for you afterwards to see the final routes.
Thank you. 現時点でマキアで、合成ルートは提示しないように作っておりますが、マキアと別でスパイアというソフトウェアがございまして、そちらでは合成ルートを確認していただくことができます。で、ルートそのものはマキアの、プラットフォーム上で見られないんですけれども、スパイアのアルゴリズムの一部が搭載されていますので、例えば、この中間体をできれば通って合成されるような分子を作ってほしい。で、こういった、ケミカルリアクションを作ったものとか、購入するビルディングブロックの価格はどのぐらいっていうのを、ジェネレーターを走らせる前にあらかじめ設定することができますので、出てきたスコアのおおよその合成実現可能性はスコアとして見ることができますので、ルートそのものは見れないんですけれども、生成されてきた分子の、どれをより合成に持っていけそうなのかという点で評価いただくことは可能です。で、ちなみにこのスパイアは2週間無料トライアルご自由に行っていただけますので、もしご興味ございましたら弊社のウェブページ確認いただけたらと思います。みたいな。
Thank you. Okay, I think you have one more question in Japanese.
No, he corrected the typo.
Thank you. So now we go back to English questions. Which docking methods are applied in your workflow?
Right now we use
Okay, right now we use Vina and RDoc. Yeah, those two.
And again, with your external API, you can plug your own. You can plug your own docking software.
Yeah.
Thank you. Next one. Does your reaction database include CH functionalization and molecular skeleton editing types of synthetic strategies?
So yes, we use everything which has been published in patents. So since that in patent there are some CH activation, it is possible to use it either in Maca in the GenAI approach or in the retrosynthesis one.
Thank you. And I think now we will answer our last question, and the last question is in Japanese. So,
yes.
Tsuki.
Yep. The question is building blocks that were used to generate the molecule shown.
Like in Makia, you mean? I guess that if we are showing the building blocks, yes, we are showing the building blocks that Makia is using to generate your molecules and the intermediates. Because remember, with Makia, it's not only one step, you have multiple reaction steps. So you can have one building block, one intermediate, another building block, another intermediate in order to arrive to your final molecule.
Thank you. 生成に使用された分子を失礼しました。ビルディングブロックを確認いただくことはできます。で、マキアが分子を生成する際に、マックス4ステップまで、反応を追加できますので、もし例えば最大の4ステップで何かを生成したっていう場合には、最初に追加したものから、最後の段階で追加したものまで全て見られるようになっています。
Thank you, Mitsuki. And with that, we've reached the end of our webinar. Thank you very much to all attendees, to our presenters, and for staying over the hour in a really good discussion. You will receive the link via email, and please make sure to fill in our satisfaction survey after the webinar.
Thank you. Bye-bye.
Thank you very much. Have a nice day. Bye.
ありがとうございます。
Goodbye.